Hi, everybody, and welcome to Neuroscientists Talk Shop, UTSA's neuroscience research podcast. Today is October 16th, 2025, and we're talking with Sara Swinford-Jackson. Sara is Assistant Professor of Behavioral and Cellular Neuroscience in the Department of Psychological and Brain Sciences at Texas A&M University. Sara's lab studies brain substrates of substance use disorder and motivated behavior. She uses rodent models for behavior and for optogenetic and genetic methods to sort out the brain mechanisms responsible for the things she's studying. Hi, Sara.
Hi. Thank you so much for having me.
Also with us, Matt Wanat, local substance use disorder guru and all-time podcast all-around star.
Howdy.
And Alexey Soshnev, our histone biologist and epigenetics expert.
Hi.
Alexey. And Ashley Miller, a PhD student working with Matt and Alexey. Hi, Ashley.
Hello.
Great. And I'm Charlie Wilson. So Sara, I know this isn't the only thing you're working on, but I think what we're going to be talking about, at least part of the time, is about epigenetics and substance use disorder. So I'd like to start the conversation with partly with a kind of brief outline of heritability of substance use disorder, which I don't think is like super well known, and then why we think there's an epigenetic component of that. And I guess there's a possibility of non-inherited, inherited, and inherited epigenetic components, and you could try to sort that out, but I think we're mostly interested in the inherited ones.
Sure. Right? Yeah, so...
And how much, why should we even suspect epigenetic inheritance to begin with?
Yeah, so we know that that substance use disorders are highly heritable, right? Somewhere between 30 and 70% of people who have a substance use disorder, their parents have had a substance use disorder, right? So we know there's a genetic component to substance use disorders, and we know there's an environmental component, but those don't entirely explain all of the differences in physiology and behavior that are associated with substance use.
So can I stop you for a second? Like, we know there's a genetic component of the disorder. How do we know that there's a genetic component? And it isn't all just your parents and your home.
Right. So there are some... I think this work has been done somewhat well in animal models, and there's some evidence in humans as well, right? So with twin studies and the population of people in Iceland are particularly well characterized and tracked genetically, right? So there's some really elegant work coming out of some labs there looking at the genetic component, right? So
that means we could identify some genes that we know are...
We know there are some single nucleotide polymorphisms that are important in certain genes that confer risk for substance use disorders, right? So obviously for substance use disorders, we can't always control, right, whoever tries drugs, right? So if no one ever tries a substance, they're not going to necessarily develop a substance use disorder.
If nobody ever took any of these substances, there wouldn't be any disorder.
Right. So we don't have like a perfect one-to-one, right? And in terms of parents, I mean, it's not like these like substance use disorders don't come about naturally like some other sort of neurological or other sorts of disorders by just naturally occur. You have to use a substance. So we don't have a perfect idea about this, but we do know, especially for things like alcohol use disorder, right, that there are some single nucleotide polymorphisms in the genome that are associated with risk for developing alcohol use disorder, but that's not 100% of the story, right? Environment is part of the story as well. And we know that those two things are impinged upon by epigenetic modifications that can influence expression of these genes and contribute to how the environment of parents can and how those exposures in parents can actually influence their offspring as well.
So is there an epigenetic equivalent of the twin study that allows you to say this proportion of it is epigenetic as opposed to just genome?
So I wish we had that in humans, right? I think that's part of the reason that folks like us are doing the kind of work we're doing, because we don't have the ability to kind of isolate those factors in humans. But we can do that in an animal model, and we can control for the genetic component and control for the environmental component, and really just isolate what's happening at an epigenetic level that might be, I think of this as more of like a modulatory role, right? So there's all of these things are going to act in concert, right? So you're not going to just see an effect of epigenetics in humans, that it's going to be not dependent on the effect of genetics and environment as well, right? So, but I think it's really some folks are looking at epigenetic marks in like human postmortem brain studies, but we're really kind of limited in how we can do that in human work. So I think that's the importance of doing this in animal models, where we can control some of these factors to really isolate the epigenetic contribution.
I guess it opens up the question of how are you actually doing that in animal models? Because, are there good animal models of substance use disorders? How can we even attempt to tease apart these various aspects? I mean, you brought up how it is multiple components can get in there. How can we really tease apart what is sort of an epigenetic component that might be contributing to problematic drug-dependent behaviors in offspring?
Yeah, so we've done this in the lab by so most of the work that I've done, there's lots of people who've done this these kind of studies, and what happens when moms are exposed to substances. Most of what I've done is focusing on what happens to dads, and the advantage in looking at what happens when dads take a substance is that in animals we can take advantage of the fact that dads play no role in their offspring's lives naturally, right? So in male rats, they don't actively parent their offspring, right? They're just that's solely the role of the mother. So we can kind of take advantage of the fact that males are contributing material genetically and epigenetically, but they're not playing any sort of environmental role in those offspring's lives. And we can use, by using rat models, we can specifically say this rat will get a drug, right? And this rat won't have access to a drug. So we're not looking at individual differences in terms of like genetic components that might predispose them to drug intake. In animals, we can say this animal will receive drug, this animal will not. And then when we train those animals to self-administer a substance, we can then mate them with naive females to produce offspring that have precisely controlled environments, right? So there's no difference in the kind of environment that they live in, or how their mom acts towards them, or what sort of exposures they might have in their everyday lives. Unlike humans, right, who are going to have all sorts of environmental factors. In rats, we can at least normalize that across our animals that receive, where their dad had received drugs, or their dad had received a control substance, like just saline infusions. And then we can look at what happens in the offspring that must have come through some sort of epigenetic modifications to the sperm, because we've taken away those genetic and environmental contributors. So to really isolate that epigenetic component.
In short, what do you see?
So we have some really interesting findings. So my interest and the work of my mentor, Chris Pierce, we've always looked at psychostimulants. So his lab had really shown first that when dads self-administer cocaine, their male offspring take less cocaine, and they're less motivated for cocaine. So they have this cocaine resistance phenotype, which sounds like it would be great, right? And sounds like that would maybe be a reason to suggest people take cocaine, but that's not actually, that's not the case, right? Those offspring also have other sorts of behavioral deficits. They have anxiety-like behaviors. They have spatial memory deficits. They're not, it's not just a universally protective thing, right? So you're not going to protect your offspring by taking cocaine. And then my work has really extended upon that to look at methamphetamine. So when, which is a related psychostimulant, right? So they're structurally a little bit different, but they both act on dopamine systems. They're both psychostimulants. But methamphetamine, actually, when you give the sires or the dads access to methamphetamine, their male offspring take more methamphetamine, and they're more motivated for methamphetamine. So they actually have this susceptibility to methamphetamine self-administration. And this is really unique to male offspring. So when we look at the female offspring, we see no behavioral changes relative to controls. So females have maybe some sort of protective mechanism. They don't experience the same sort of effect of paternal drug self-administration on their behavior in adulthood.
So I'm wondering these changes here, and maybe you haven't, or nobody's done these experiments yet, but you were giving the sires one drug, so cocaine, and then you were testing the offspring with cocaine. And I mean, if one of the things we've sort of seen with the over the years is that often the quote unquote popularity of what is the problematic drug, it changes over time.
Right.
And I guess, have you looked at these changes here? So in the males of the cocaine, whose parent was taking cocaine, they're taking less cocaine. Have you looked at those who had cocaine, the parents had cocaine, or the dads, and then looked at methamphetamine or opiates, or, how global are these changes? Is this sort of drug specific, or is this, maybe, functional changes? Maybe the males that were taking less cocaine, maybe they just didn't learn the cue outcome association as well. And, are they going to have impairments in taking all other drugs, or vice versa?
Right.
And I guess.
Yeah. So one of the things that we often look at when we're looking at drug self-administration as a control is the non-drug reinforcer sucrose, right? So you really want to evaluate whether there's differences in reward and reinforcement that are drug specific versus all sorts of rewards. So for both cocaine sired and methamphetamine sired animals, there's no difference in the amount of sucrose that they take. So they respond similarly for non-drug reinforcers. We haven't done the kinds of studies that you suggest in the context of cocaine. So we haven't looked at offspring and how much they self-administer other types of drug in the context of cocaine, but I have done that with methamphetamine. So we looked at when dads self-administer methamphetamine, when we look at cocaine self-administration or morphine self-administration in the offspring, we see no behavioral changes. So they self-administer just as much cocaine as control animals. They self-administer just as much morphine as control animals. So it suggests it's not a dysregulation of the reward system or reinforcement, broadly speaking, and it's also not an enhancement necessarily in learning or deficit in the case of those cocaine resistance animals. That's an important control, right? If you see a decrease in intake, you want to make sure it's not because they don't learn as well or they struggle with some aspect of the behavioral task. But we really seem to see, at least in the context of methamphetamine, I wish I could tell you more about cocaine-sired offspring, maybe in a future time, right? But at least in the context of methamphetamine, it seems to be that we have a specific increase and a specific susceptibility to methamphetamine self-administration in those offspring, and that doesn't extend to cocaine, which was surprising to us. We thought maybe we would see this increase in psychostimulant use, broadly speaking. So, and it doesn't extend to morphine as well. So I think those are potentially really interesting, and I think that might say something about the mechanisms that are involved in some way. But it's hard to know, right? So I think that's one of the other things that we haven't done that I think is so important at this point is that we know that people who use substances don't typically use one substance at a time, right? When we're modeling this in animals, we are giving them access to one substance at a time, but humans who use drugs very rarely use one drug to the exclusion of other things, right? So people who use cocaine are often going to smoke cigarettes or drink alcohol or do something else. In animal models, we're really limiting them to one drug at a time. So it'd be really interesting to kind of see how this influences, do they take more methamphetamine at the exclusion of other kinds of drugs if we gave them access to multiple kinds of drugs? That would be a really interesting thing that we haven't done yet, but I think that would kind of give us more insight into how susceptible these offspring are.
It seems like a lot of clues.
Yeah.
And so I'd like to try to imagine what kind of epigenetic mechanisms that we know about would be consistent with that. I'm trying like build a scenario of how taking methamphetamine long time before you conceive a offspring could alter the offspring's susceptibility, and what could be the intermediate biology that carries that information. So what do we know? I mean, it could be hypothetical if we don't know everything, but I'm kind of interested in, because I don't have a really clear intuition about how epigenetics works.
Do you want to take that, or?
Sure. Well, so I'll say that in this model, we're really isolating the only contribution that sires can make to their offspring, right, is through contributing sperm. So we know that there has to be some sort of drug-induced epigenetic change in the sperm that is contributing to changes in offspring physiology and offspring behavior, and there can be lots of different epigenetic modifications. So we can look at histone post-translational modifications. Even though histones are mostly replaced by proteins, there are some retained histone modifications throughout spermatogenesis that could be important. We could look at things like DNA methylation or microRNAs. We've kind of done a little bit of work looking at each of these in sperm and in individual cases to kind of start to understand what are the epigenetic modifications that occur consequent to drug exposure.
Does it have to be on the Y chromosome? I mean, that's the fascinating thing is you're getting all of these these alterations in behavior that are only in the male. So it almost screams it's got to be something on the Y chromosome. Is that it, or is it more complicated?
I wish it was that simple. It appears not to be. So the where we found changes don't seem to be specific to just X or Y chromosomes, right? I think that, I wish we had a really good example of why we have a sex-specific phenotype. I think that's more complicated, and I think there's something, my hypothesis is that there's something about female biology, especially maybe early in development, that's sort of protective, because these females don't seem to have these behavioral deficits, and they also don't have the sort of changes in gene expression consequent to these epigenetic changes in sperm that we see in male offspring. So they're intact, or at least the same as controls, both behaviorally and physiologically. So we have a couple, I think, your question about how this is happening, I think is a really good one, because I think we have a couple of kind of black boxes in this process, right? So we know that drugs are producing some sort of epigenetic modification. This is happening in sperm, but how drugs like cocaine and methamphetamine are doing this in sperm, I think, are really not necessarily clear.
So they bind to receptors on some cell in the testes, right? It's got to be something like
that. Yeah, so the second highest binding site for cocaine, and I think methamphetamine as well, but certainly for cocaine, so if you radiolabel cocaine, it binds to the brain, and then the second highest binding site in the body is in the testes.
And those are in developing sperm or in some supporting cell in the testes?
So it's a good question. We have done some studies, so we don't exactly know like how drugs are inducing those epigenetic modifications, but we have done some studies to kind of tease apart where in this process drug exposure is important. So we basically have asked the question, if your dad took drugs in college, right, or your dad took drugs right before you were conceived, does that matter?
Yeah.
And I think that's a really important question, because that's relevant to lots of people, right? Especially not just in the context of drugs like psychostimulants, but in the context of drugs like alcohol and nicotine as well, right?
And all kinds of epigenetic stuff.
Right. Yeah.
Including positive things.
Yes.
Not just negative.
Yes. Yeah. So we've done kind of a strategy to change the recency of drug exposure relative to when those offspring were produced. So we typically, when we do this, we expose the sires to a drug for the duration of spermatogenesis, and then we mate them right away. So we're looking at sperm that have the potential to have been epigenetically modified throughout that period of spermatogenesis. and then we look at those offspring. But when we, to kind of look at how long that effect persists and kind of to get at the question of whether these spermatogonial stem cells might have been modified, we did this delayed mating paradigm where instead of waiting, 1 day and kind of mating them right away, we mated them at 1 day and then again at 45 days and then again at 90 days. So that's a cycle and a half of spermatogenesis, and those sperm have been washed out a couple of times by breeding, by matings in between. And when we look at offspring produced after 90 days or a cycle and a half of spermatogenesis since the last time the males had cocaine or methamphetamine, we see the effect disappear. So it seems like this is not a long-lasting, permanent change, right? It seems like there's some sort of vulnerable window for epigenetic editing during spermatogenesis, and everyone can breathe a little easier that their decisions a long time ago might not necessarily directly impact their offspring, at least in this context, right? At least in the context of drugs. So it seems like there's some sort of direct effect, and it's not. You're not destined to have these effects last forever.
So you mentioned, it's fascinating that cocaine, binds to the brain and binds to the testes. Is there any way to sort of figure out, which one might be more important in sort of mediating these epigenetic changes? Are there any tools that, we can do to figure out, is it acting in the brain, testes, or both?
Yeah, so we have actually done, we've kind of tried to answer this question in the context of cocaine. So we've used an analog of cocaine called cocaine methionide, which doesn't cross the blood-brain barrier. So in this way, cocaine is able to presumably get to the testes, but doesn't get to the brain. And animals won't self-administer this, right? Because it's not gonna provide any of those reinforcing or rewarding properties. So we've delivered this in a yoked manner. So that means every time an animal gets an infusion of cocaine, its partner gets an infusion of cocaine methionide. And then when we mated those animals to produce offspring, they still also show this cocaine resistance phenotype. So it suggests that maybe brain cocaine is not necessarily important, but maybe cocaine binding somewhere else, maybe the testes. We think the testes, right? We didn't specifically direct cocaine to the testes, but we did at least prevent it from getting to the brain, and that suggests that there is some sort of, this is probably mediated by peripheral effects and direct effects maybe of drugs in the testes, and maybe not necessarily in the brain. Maybe in the brain, maybe volitional drug use is part of this, and it might influence some phenotypes in the offspring, and certainly may have different epigenetic modifications, right? But at least it suggests that the main phenotype isn't necessarily driven by brain levels of drug and maybe direct levels of drug in the testes.
So is that the same then for meth? Do we know where meth is binding? Is it the same thing? And then would we predict the same thing for opiates and other drugs where, you're getting these selective effects in the males? Well, now this sort of makes sense. Okay, if it's maybe the testes are sort of the, ground zero where these changes are occurring, how do abuse substances potentially interact with the cells there? And are there some similarities or differences in sort of how these, one cyclostimulants and, different classes of drugs that they might interact?
That's a great question. I wish I had a more concrete answer for you, right? So I haven't personally done any of the work with opioids in this context. They are also able to induce epigenetic modifications in sperm. Opioids are an interesting case, right? Because you have opioid receptors in other places, right? And they're different. They're very different than psychostimulants, right? They're not going to act on dopamine transporters necessarily, right? Or serotonin or norepinephrine transporters. So it's certainly, I wonder, right? If there's a common mechanism somehow, or if the mechanisms are very different, but drugs are exerting these peripheral effects that we don't necessarily fully understand. And I certainly am very interested in how peripheral binding of drugs to the testes are actually inducing these sort of epigenetic modifications. But I don't have a good answer for that, and I really I wish I had a better answer for how these epigenetic modifications in the sperm are changing the brains of offspring, right? So, and how they're doing that in a sex-specific and maybe region-specific way.
When you say germline and when you say sex-specific effects and you say epigenetics in the same sentence, and it's invertebrate model, so placental mammal, the first thing that comes to mind is imprinted genes.
Mm-hmm.
And the imprinted genes are more than just, a couple loci on the X chromosome. It's, hundreds of imprinted loci. It depends who you ask, but canonically about 200 imprints in
Can I get
a
a
mouse.
definition of imprinted?
So these are loci that, let me take a step back. So we all have 2 copies of every gene, except for the sex chromosome genes. 1 copy comes from mom, 1 copy comes from dad. And what happened in placental mammals specifically is the dad wants the kids to be big and strong. And the mom wants the kids to be small because of the birth canal situation. So mom wants to suppress certain growth at certain times, and the dad wants to increase the growth.
These are not really thoughts they're having.
They're not the thoughts they're having, but that's the biological reality they're facing.
Yeah.
And so what happens is certain alleles, not all, but a few hundred genes inherit not just the DNA sequence from the parents, but also the expression pattern from the parents. So the copy that comes from dad will be expressed highly, the copy that comes from mom will be suppressed, or vice versa. There are different types of imprints. Now, normally that all works well and everything is good, but sometimes these imprints get messed up, and that's when you have childhood overgrowth syndromes, for example. A very famous one is Beckwith-Wiedemann syndrome. There's a few others where multiple developmental defects occur because of that, but at the end of the day, they're tied to changes in growth that occur because, let's say, both alleles are expressed as if they come from dad. Like, that's an example. That is typically connected to DNA methylation. It doesn't have to be, and it's actually quite complicated story. So, yeah, there's more to it than that. But now that you say that, okay, we have this effect that gets inherited paternally, and that gets expressed in male offspring only. I am very curious about imprinted loci, and if there is any evidence or interest or way to look at what happens to paternal imprints in the offspring.
Yeah, we have not done that in a kind of systematic way, right?
Mm-hmm.
One of the things that I guess I don't know, maybe you know, so one of the things that I find kind of interesting about this is we also see regional specific changes in gene expression, right? So it's not that we have expression changes throughout the whole brain or the whole body. We see expression changes specifically in the nucleus accumbens, for example, or specifically in the case of like spatial memory deficits, in the case of in the hippocampus, right? So it kind of points to maybe that's not, you wouldn't expect, right, imprinted genes to have like region-specific patterns of expression. Or
I don't know if
differences in
anybody
expression.
knows.
Yeah.
I think it's such a evolving topic,
Yeah.
and especially when it comes to brain. I mean,
Yeah.
everything we know about brain, we are basically rewriting in the last
Yeah.
5 years
Yeah.
because of the explosion of single-cell sequencing.
Yeah.
So
It
is it a developmental thing, or is it maybe there's a mosaic sort of or yes or no?
Yes.
Okay.
Yeah.
It's absolutely a developmental thing because these imprints are active at different developmental timings, but then they're also clonal. So there's absolutely clonal growth, and especially when it comes to growth paradigm, let's say in a very early development, a group of neurons gets some growth advantage, they will outcompete the surrounding neurons, and so then you have a instead of like a normal mosaic where everything is represented equally, you have a single clone that took over a whole region in the brain. There are a lot of people who are studying somatic mutations in the brain, and it's a really fascinating field these days. I don't know enough about it to talk about it.
But these imprinted genes are, it kind of sounds like they're very restricted in function to just growth, but not necessarily.
Not necessarily. There's about, so in the mouse, the overall number is roughly 200, which 200 genes is 1% of the whole mouse
So it could be
genome.
ones
That's
that
not
even
bad.
have to do with risk-taking or something like that. I mean, maybe dad wants his offspring to be adventurous and mom would like
There we go. Or aggression
Yeah.
behavior, right? Like what we
So they-
typically associate aggression with male territorial behaviors, like mouse for sure, rat
So,
for sure.
Yeah.
so somehow, I mean, we know that cocaine changes gene expression enormously. And there's a big long list of genes that get changed in
Mhm.
regulation by cocaine. So I'm guessing something similar to that happens in the germline. So those cells change expression of something because they have had amphetamine. Of course, it's really dopamine that's responsible for amphetamine-induced changes in expression in the brain.
But we don't necessarily know if that's really the. I think we don't know enough about what happens in the. I don't know enough about what happens in the testes to say.
So maybe
it's certainly
it's possible amphetamine and cocaine aren't binding to a dopamine transporter, but to some other target that we don't know about. But we would normally think of them binding to a dopamine transporter.
Right.
Well, they've made dopamine transporter knockout animals. Has anybody, and so we're going to mice,
Yeah.
but has anybody looked at these types of questions using these or overexpression models? Because this could get at the...
Yeah. I think that's a great idea. I haven't. I don't think anyone has. But
That's
I think
great.
that, yeah. I think that would be a really interesting way of approaching this, right? So to try to see, what is it the principal actions that we think of for these drugs in the brain? Are those really what's important in mediating the peripheral effects? I think that would be an interesting question. We haven't. I will put the caveat out there that we haven't done these studies in mice, right? So some of these things are a little tougher sometimes to do in mice than they are in rats. So I presume we would see cocaine resistance and methamphetamine susceptibility in mice as well. But it would be really interesting to do that in the context of a knockout, or in the context of a region specific knockdown of DAD or SERD or NET in the testes.
Or the flip way, cocaine's used medically as an anesthetic. I mean, you can go back to dentistry back in the 1800s. This was, hey, lidocaine. That's a way that you could potentially, I don't know, low-key way of being able to test those ideas.
Yeah, absolutely.
So I don't want to let you get away without talking about inheritance. So the rats whose father was exposed to cocaine had to be exposed at that time of spermatogenesis. So that's kind of maybe good.
Yeah.
But then that animal has an epigenetic change that is forever. Right? And then that animal's offspring may also be affected. Could you say something about how far this goes? How many generations down can this go? And what's the pattern of generational transmittance?
Yeah, so it depends on the phenotype that we're looking at. So we have taken this to the F2 generation in the context of cocaine, and in the context of methamphetamine. One of the things that I think is really interesting. So when we're doing this, we're mating F1 males, right? So the offspring that have been produced from dads that have self-administered methamphetamine. But these animals never take methamphetamine themselves, and we just mate them with a naive animal to produce an F2 generation. And we've looked at methamphetamine self-administration in those offspring, and we actually see no difference in male offspring in this F2 generation, or grand offspring, right? But we do actually see a potential an increase in female offspring and these grand offspring. So somehow, even though these female F1 offspring were not affected, the F2 generation, there's maybe some sort of mechanism by which transmission is selective for females, so we actually see them take more methamphetamine than their control animals. This is really puzzling, so we kind of went back and said, okay, is there something maybe latent in these F1 females, right, who don't have any difference in methamphetamine self-administration, and do they, is there some sort of change that also occurs at the F2 generation after we mate them with naive males? And when we did that, we found no differences. So they seem to have no difference in the F1 generation for females, and their offspring are also not different. But there's something that happens in the F1 males that confers susceptibility in the F2 females. For me, I think this is really interesting. It's similar to some work that we reviewed today, actually looking at heroin intake in F0s, and they actually also see this pattern of increased intake in the F1 generation in males, but increases in heroin intake in the F2 females, but not F2 males. So I think that's potentially really interesting. I don't have a good explanation for that, right? Because we haven't started to profile like what happens in the sperm, for example, of those F1 animals that would be transmitted to those F2 females. But there is some sort of transgenerational susceptibility. It seems to not be, there are other sorts of phenomenon that have maybe more long-lasting, multi-generational impacts. This seems to be, at least it doesn't continue right along multiple generations of male offspring, which is also kind of reassuring, right? So, especially given your example of like when people are using cocaine medically for lots of reasons. But I think that's really interesting, and maybe provide some insights into maybe that investigating that can provide some insights into why we see a sex specific effect in offspring, but we haven't done that yet, and I think that'd be really interesting.
And we don't know if it's finished yet. I mean, the next generation might.
We don't know. I haven't continued to mate them to see exactly when it stops. I think that would be really interesting. It's very confusing, right? To figure out how you go from an effect in males to now a specific effect in females that you didn't have before.
These are also studies that require an awful lot of patience.
They do.
And that's the problem with a lot of epigenetic inheritance studies, that you would take 2 grad students to get to the bottom of it, and so somebody spends 3, 4, 5 years,
yes,
and then has to hand the project over. It takes generations of graduate
so
students.
they inherit the project.
Yes,
so they have
exactly.
Yeah.
So you can't just blame your parents. You've got to blame your grandparents as well for who you became. Okay.
And your lab parents for making you do these kind of studies, right?
The sins of the father are visited upon the sons for 2 or 3 generations, except it may switch to the daughters.
Yeah. So there are so many things, right? I think there's so many interesting things about this project. I think that's one of the things I find so fascinating about it is there's a million different ways you could go. There's a million different potential behaviors and potential impacts on physiology in the offspring that you could kind of try to understand, and how those all interplay together to influence behaviors like drug self-administration, and how those overlay on top of the genetic and environmental components kind of really speaks to the absolute complexity of the kind of research we're doing, the kind of systems we're working in, and how all those things kind of coalesce.
That's a great summary. And maybe it's a good time to stop then. So thank you, Sarah. I'm sorry, Ashley,
Oh.
you want to say something?
I did have one question. We were using the terms drug exposure and self-administration almost as if they were the same thing. But oftentimes when we're discussing the epigenetic inheritance of effects from drugs, I think it's important to make the distinction of, is this an effect of just exposure to the drug, or especially when we are talking about something that could be cerebral or more peripheral, could the cerebral side require more of the animal has to take it themselves? There has to be that self-administration, that voluntary aspect to it. And when you remove the contingency, would you see that across both the cerebral and the periphery?
Yeah, I think that's a really good question, right? So, and maybe kind of a unique feature for animal models, right? Because we kind of don't do this in humans. We don't like give them drugs that they have no control over. There are some studies that have looked at kind of tried to understand the motivational aspect of drugs and how that might influence their offspring. The converse of that is that they will give drugs non-contingently and basically pair animals so that for every infusion that an animal self-administers, their partner gets a yoked infusion. Animals find that stressful, right? So they don't really enjoy kind of getting non-contingent drug rewards whenever someone else feels like it. So they find that a little stressful. It's a little bit hard to separate, but I think studies like the kind of like cocaine methionide study that we did at least suggest that the peripheral component is enough to at least recapitulate some of these behaviors, but maybe some of the exact epigenetic mechanisms that are influenced by the other components of this process, right? Like the feeling of reward and the actual drug intake and seeking the drug and intentionally taking that drug may also be important and may have different effects than just drug exposure themselves. So I think in some cases they may be similar, but in other cases, potentially pushing the animals to increase how much drug they take, or asking questions in ways that we model other features of addiction, right? So we know that people who have substance use disorders take drugs despite negative consequences, and they have increased motivation, and they have compulsive drug taking. And if we kind of model some of these aspects as well, beyond just the pure drug intake, we might kind of get at more of those like ethologically relevant components of this as well. So I think that's an important point. I think there's always more. Fortunately for all of us, I think there's always more work to be done in this area.
Thank you, Ashley, Alexey, Matt, Sarah. This has been Neuroscientists Talk Shop.